Hey guys, I'm Dr. Robin and I'm back for a new episode of K-Pop Science EDU. To celebrate the fact that I'm going to see Epik High in Boston this Saturday, I thought it would be perfect to do a science lesson related to them. I was brainstorming ideas, walking around singing It's the Elements, and my friend Katie suggested I should do a lesson about the science of strawberries because Epik High's new album is called Strawberry. I thought this was perfect because I often do an activity with my science club in which we extract the DNA from strawberries. And as we all know, extracting DNA from strawberries is a lot easier than you probably think. First off, there is a lot of DNA in strawberries. Second off, in addition to this being a cool lesson for everyone to learn, this is a great activity to do with kids. There's kind of a nice balance of controlled mess, you get to mix chemicals together, there's some potential ickiness. Now for fun, I made these cool business cards, which I am way too proud of how cute these came out, that have the protocol on them. I'm going to pass these out to people at the concert. And I will also go ahead and put a link in the YouTube video's description. So if you want, you can download and print them out. So what is DNA? DNA is often referred to as the blueprint for life because the DNA in our cells contains all the instructions our cells use to make everything our bodies need. Now, since my PhD's in genetics, I obviously think DNA is really, really cool, and I could go on and on and on, but I'm going to save that for other videos. For now, I will simply say that 1, living things are made up of cells, and this is a drawing of a generic plant cell, like a strawberry. 2, most cells have DNA in them. 3, DNA contains the instructions cells use to make everything living things need. And 4, the DNA is in the nucleus. And what does extracting mean? Extracting DNA means getting the DNA out of the cells. In order to get the DNA out of the cells, we need to bust open the cells and we need to bust open that nucleus where the DNA is. Now in a lab where your samples of DNA need to be really clean, you can buy special kits and they give you all the chemicals and you can have a really good, nice DNA sample at the end. But really, household products do a pretty decent job. So let's check out the protocol. The first step is to crush 2 strawberries in a Ziploc bag. This step breaks down the tissues of the strawberry so that we separate as many of the cells from each other as we can. We want to be able to expose as many of the cells as possible to the chemicals we're going to add. Okay, so let's give that a go. Just have a regular Ziploc bag. Now it's not strawberry season. These are actually some pretty tough strawberries. My practice run of this was more difficult than I imagined, but I'm just gonna smash them up as best you can. Okay, so here is our mashed strawberry. Then we make up our DNA extraction liquid by mixing 2 teaspoons of detergent with 1 teaspoon of salt and 1/2 cup of water. Okay, so 2 teaspoons of detergent. One teaspoon of salt. And then 1/2 cup of water. Cheating a little bit and using a solid 1/2 cup for this because I just thought it was easier. That's fine if you want to do that. All right, mix it up. Now we add 2 teaspoons of this to our strawberry mash. Depending on how brave you're feeling, you don't have to reseal the bag, but like I said, it can get messy. Mix it all together. Now remember, you want to really break apart any chunks of the strawberry because you want to expose them all to the detergent. So what does that extraction liquid do? Cells, both animal cells and plant cells, are surrounded by a cellular membrane that keeps cell parts in and non-cell parts out. And inside the cell, a membrane surrounds the nucleus to keep the nucleus parts, including the DNA, inside the nucleus. In order to break down these membranes and get the DNA out, you need a chemical that will push holes into the membranes. It turns out that soap and detergents do just that. This is actually how soap and detergents work. So once we mix our strawberry mash with the detergent, we end up busting open the cells and the nuclei and ending up with a whole bunch of cell parts mixed in with the water in the bag. Now the DNA and the salt dissolve in the water, but the other cell parts do not. The next step is to filter out all the debris by pouring the mash through a coffee filter. Now in order to do this, it kind of depends on how many hands you have. So I usually use these bucket coffee filters and put it in the top of a glass. Now, if there's enough of you and someone can hold the filter down, that's great. What we usually do is we put the filter in the top and then adhere it with, actually usually use 2 rubber bands, just because I had some moments with 1 rubber band. Okay, so we're going to pour our mash into the filter. And the DNA and salt is dissolved in the water, so when the water passes through, the DNA and salt should pass through as well, and the rest of the chunks of strawberry will not. Now, as you can note, it's not filtering very quickly. I didn't do a great job smashing up the strawberry, so that would help, but in general, we do often need to give this a little help. So in order to help it along, this is where the controlled messiness can come in. I would suggest sliding down the rubber bands if you try to lift them off the top. Usually accidents happen. And you're just going to kind of press on this to help the liquid out. So the water with the DNA and salt is filtering through. Now you can't squeeze it too much because, you know, just a coffee filter. All right, I think that's gonna be enough. Have some paper towel nearby. All right, to then get the DNA out of the water, we mix in 1/2 cup of ice cold isopropanol, otherwise known as rubbing alcohol. Now the DNA is not soluble. In other words, it does not dissolve in the rubbing alcohol. And the salt we added before binds to the DNA and neutralizes its negative charges. So together, these 2 things mean that the DNA ends up clumping together in a process called precipitating. So through this process, we end up with long strings of strawberry DNA. Okay, so let's try it out. Here we've got the ice cold isopropanol. All right, now swirl it around, take these rubber bands off so they don't interfere with stuff. Okay, so I think you should be able to see this. There was no solid in this before. So as you can see, does it help if I put this behind? Yeah, I think so. And all of that stringy stuff is the DNA. So you can pull it out of there, the toothpick. I'm gonna use a toothpick, but you could use Q-tips, probably a fork. Actually, a fork's probably a really good idea. I've never done that. Put it into a vial, put it into a cup. I recommend, you can get these from Amazon, probably a lot of other places. I'll put a link in the YouTube video description. These are Epik tubes, and this is what we really use in labs. Like I've probably used 8 million of these in my life. And they're just a lot of fun because it just, you know, you pop them open and it's real science materials. So I think this would be great. They're not too expensive. You can buy a bunch of them. Keep your DNA in it. So let's see. So scoop it out. Look at that. That is all strawberry DNA. So let's see. Usually the hardest part is getting it off of the toothpick. See if I can do this. I usually end up using my fingers, making a mess. This is more of the mess part. So I'm gonna try to use 2 toothpicks. See if I can do this. Perfect, all right. So this, I think this helps, maybe. So we've got, there's a little bit of liquid in the bottom, but all of this is DNA. So that is your strawberry DNA. It's so cool. And that is it for today. If you do decide to extract some strawberry DNA, I would love to see some video or pictures of your fun. Until next time, keep listening and keep learning. Kamsahamnida.